The broadcast is now starting. All attendees are in listen-only mode.
Good afternoon, everyone. Can you hear me all right? If you can hear me, please leave a comment in the chat box. Can anyone hear me all right? If you can hear me, please leave a comment in the chat box. Okay, great. Excellent, excellent. First of all, thank you for joining our webinar today. Before we begin, let me give you a brief introduction. My name is Ming Zhou. My background is in chemistry and I have been with MBA Scientific Consultants for 5 years. MBA Scientific Consultants is an analytical microscopy laboratory. We offer customized analytical testing, investigative analysis, and ongoing R&D support. We have been in business for 26 years, and our mission is to create value by providing unique analytical solutions and technical data interpretations to help clients solve problems. Our speaker for today is Rich Brown, which is an executive director and a senior forensic microscopist with MVA Scientific Consultants, which has been with MVA since 1991. He specializes in investigative analysis, method development, and material analysis, which has a master's degree in forensic chemistry from Northeastern University in Boston, Massachusetts. Now I'm going to hand it over to Rich.
Thank you, Ming.
You're welcome.
If everyone can see my screen, our schedule for today, our outline is I'll talk a little bit about my rationale for sending a sample out, that is to another laboratory. I'll talk about particle characterization and the complementary techniques that I use here at MVA. I'll talk about a few case studies, and then we'll have some time at the end for people to ask questions, which is your time to really try to stump me. As far as the rationale, if I have to send a sample out, and I do that occasionally, I usually have an expectation of what type of analysis I'm going to get from the laboratory I send it to. So it's always good for me to have a discussion with that laboratory, one, to give them a background about the sample, about my expectations, and anything that would help them in their work to get to a conclusion for this investigation. So that communication to me is very important. The second is I like to be kept apprised of the progress or the lack of progress of their analysis of my sample. There's other things going on constantly, but it's still nice to have an update saying, hey, we just got a chance to look at your sample, or this is what we're finding. This is preliminary data, and with that is a big caution because that data can change as the analysis proceeds, and I certainly expect that. And then finally, if I don't hear from the lab, if they tell me it's going to take 10 days or 2 weeks, and I don't hear from them, I'm going to give them a call and say, hey, what's going on with my sample? Where are we? Is there anything I can do to help move it along? So that type of communication for me is very important when I send a sample out, and I would assume it is to most of the people listening today. Finally, I'll get a report, and at that point, I'll have a chance to read the results, go through the results, and at that time, I'd like to call the lab again and say, I've looked at the report. Can you explain or clarify this point, or is there anything else as we go through the report, or just discuss general terms what the report means? Does anything else come up where we have to do more work, or are we done? Do any additional testing or additional tests come out of that conference? All these things are very important to me as I investigate a particular problem. In the image on the screen we have now, when we talk about contamination, and that's irrespective of the industry that the contamination occurs in, something's out of place. We don't always find ice machines on the beach, but in this particular image, there's clues. I took this picture in Florida. There's clues as to what happened. There was a big concert the whole weekend prior to me finding this on the beach on Monday, And if you could look around on the surface of the sand, you can see the poles that were used for the fencing. So the machine was put there to help with the concert venue itself. So that makes perfect sense, but still, it's not something you find on the beach every day. An explanation of forensic industrial investigation, which is what I primarily do, is the application of analytical techniques to determine the cause of product failures, defects, and product returns for manufacturing. The types of samples and industries that we serve that are in-house now, we have pharma, drugs, medical devices, drug delivery systems, chemical manufacturing, anywhere from fillers to fertilizer. We work for aerospace, we look at coatings, we look at engine components. You're not? Okay, I apologize. It looks like my screen wasn't being seen. Can everyone see my screen now, the industry served? We're good? Okay, I apologize for that. We missed a couple of slides. But essentially, we're talking about the industry served. Construction materials, every different type of construction material I can think of, we've looked at. Consumer goods, I think we're done with the Winnebagos finally. And environmental dust, nuisance dust, where do they come from, what's in them, things of that nature. The types of techniques we use, polarized light microscopy, or PLM. We have a light microscope with an analyzer and a polarizer to look at transparent samples. We have Fourier transform infrared microscopies, with essentially a microscope with a Fourier transform infrared spectrophotometer attached. Scanning electron microscopy with energy dispersive X-ray spectrometry. We actually have duplicates of all these instruments except for the FTIR. Confocal Raman microscopy, or microspectroscopy, where we basically have an incident laser hit the sample and we look at the resulting Raman spectrum. Transmission electron microscopy, this is a mouthful. Energy dispersive X-ray spectrometry with a selected area diffraction, or AEM, analytical electron microscopy. For smaller transparent particles, electron transparent particles, or particles that can be made electron transparent through certain preparation techniques. And then we have some scanning white light interference microscope, where we can do surface roughness profiling. We also have some dark field fluorescence, microspectrophotometers, other types of microscopes in-house that we use for characterizing samples. Some examples of what they look like, for those of us that don't know. In the upper left, we have a transmission electron microscope. In the center, we have a field emission scanning electron microscope. We have Randy sitting a polarized light microscope on the upper right. Lower left, our FTIR microscope, which is an oldie but a goodie, and a confocal Raman microscope. The polarized light microscope is probably our most useful piece of equipment. Pound for pound, dollar for dollar, it's the least expensive type of microscope, but there's a trade-off in that the person using it, it's usually a minimum of 5 years experience to become proficient in it. So there is a steep learning curve on it. It would be nice if every time particle contamination happened, we were there to witness it. That's not always the case. Usually we're left with the particles themselves, and we have to backtrack through the process to try and determine what they are and where they came from, which is what we do. What are they? Where'd they come from? They need to be isolated and collected. They need to be examined. They need to be analyzed and measured using the analytical tools that I just talked about. And they have to be characterized and identified. During the process, we'd expect that we'd have a quality type of an analysis. We want it to be quick. This type of analysis involves studying the samples. It's not a simple procedure where we grind it up and shoot it into an instrument and get a chart or a chart result and then write a report. There's actually some time involved in this process. We'd like the people doing the work to be competent. We also want them to be confidential. People don't like to know that they have problems, and we certainly don't like to share problems that other people have with our other customers. The analysis should be complete. The report should speak for itself. And ideally, the lab would be close, like next door. That's not always the case. We can say that for our local customers, that is the case, and we like to help them as much as possible. But you pretty much lose a day if you have to send something by FedEx or through the mail. So you're a day behind when you start. Speak to classification versus identification. I like this slide mainly because everyone has seen these most of their lives. They know what they are. They can recognize them just by sight. And to the microscopist, a lot of small particles are the same way. If we shrunk these down and made them very, very small, we could still tell by looking through the microscope which one was the apple and which one was the orange. We can section them, look inside, we can look at the thickness of the skin, we can look at the surface, we can look at the color differences, we can look at the cross section and see that they're different. But without having an expert that could identify which species of orange this is or which species of apple, we're a little bit limited. So we can do the classification, the actual identification, what species is this, that takes a higher level of expertise. Same way with small particles. If we go through a hypothetical particle quickly, we get a particle and it presents to us as a white flake. We're looking at it under low magnification with reflected light. We see its color is white and it has a flake type appearance. If we put a cover slip on it and press on it, we can observe that the particle deforms. It spreads out. It spreads out in a plastic type of process, whereas it doesn't bounce back to its original shape. At that point, we know it's plastic as opposed to elastomeric or rubber, where a rubber you would expect to return to its original shape. So we've got some important information already, and we're about 10 seconds into the process. The white flake is an acrylic polymer. Now we've done some FTIR analysis, and we've determined that the polymer is an acrylic. The white acrylic flake contains titanium and aluminum silicon. We see particles embedded in the polymer, some of which are titanium, some have aluminum and silicon associated with them. So at this step, we have a white flake. It's an acrylic polymer, and it has fillers, pigments, titanium, aluminum, silicon. We don't know yet if that aluminum silicon is associated with any particular type of compound, or if the titanium is a particular type of pigment. So we can determine that, and one way is to low temperature oxygen plasma ash that resin away, release some of those particles, put them on a TEM grid and analyze them by transmission electron microscopy, and then make a determination that we have kaolin, clay, and titanium dioxide. So now we have a white flake. It's an acrylic. It contains titanium, aluminum, silicon fillers. Those fillers are kaolin and titanium dioxide. And the point of this is to show that particles that are aggregates or that contain different components are very useful when we try to do particle characterization, and then we try to go back to a possible source. The more facets, the more types of materials that are associated with each other, the more clues we have to backtrack through a process. They can be intimidating when you have a large number of particles, but for the most part, it's the most useful. So a typical everyday substance that we use day in and day out, magnified, no scale bars, I don't want to give that away. By looking through a polarized light microscope, we're looking with a single polarizer, bright field. We have a squarish particle, and next to it we have a dark particle. And if you look carefully at the dark particle, you can see my cursor, it looks like we have some cellular structure here. So one question is, how typical are these particles of the whole? And if we look at a little lower magnification, back off a little bit, we can see that the square particles are very typical. This is typical of this material. It's very representative, whereas on the right, the darker particles, there's all different colors, all different shades, all different shapes. So it may or may not be typical of the original material. And then finally, to give it away, it's salt and pepper, something that we use and see every day. Point being that the sample needs to be representative. If it's not representative, the analysis you do will tell you something about the particles, but it may not tell you enough about the source or potential sources. We'll go through some examples that we use for some of the workshops that we give. This is a black fiber. It's on a filter membrane that we've isolated from a product. At higher magnification, we're now looking through a polarized light microscope. With a single polarizer, we can see that the fiber is dark. It actually presents as being black. If we cross the polars, so now that we have a black background, we can see that the particle is still, or the fiber is still bright in some areas, and that's very diagnostic. It tells us that this particular particle has more than one refractive index. No, we're still transmitting audio. Okay, if everyone Ming just had a comment that we might have lost audio there for a moment. So if someone can respond that they still have audio, that would help us. We're still looking at the cross polarizers. I know this is cotton because I've seen it before. We're going to look at the same fiber. We're going to look at it by scanning electron microscopy, which is not always useful for looking at a cotton fiber, but we start to see some of the structure that's diagnostic for this type of a seed hair. We have this twisting and we have this flattened appearance. And if we do an analysis, we do an elemental analysis at this particular spot, we get carbon and oxygen, which is typical for the cellulose, but we also have some sulfur present, which may help us with the dye. That is, if we had something to compare it to, we'd expect to see sulfur if it was a similar source. By IR, we get a typical cellulose spectrum. For some cotton fibers, cellulose is a strong absorber, so this cellulose spectrum will dominate the IR spectrum. But for some that are dye, we can get some information about the dyes while it's still in the fiber without doing some extra steps and trying to extract that dye. This comprehensive approach, or this complementary approach, gives us a number of different views of the same particle type, so that as the analysis proceeds, as our work proceeds, we can cross-check each other. So that if polarized microscopy finds cotton and I'm looking at a fiber that doesn't look anything like cotton, we'll have to have a discussion and see if we're actually looking at the same fiber. So that quality control is built into the analysis. And then finally, we just compare it to a reference spectrum of cellulose. We'll be looking at the blue particle next. This one, when they compress it, it rebounds. It's elastomeric. By polarized light with a single polarizer, it's fairly opaque. We can start to see some transparency on the edges. And if we look carefully, we see some black dots that look like they might be fillers or pigment. Cross polarizers, not much information there, except that now at the edges, we start to see some particles that are very bright. That tells us those particles, fillers within this elastomeric compound, are birefringent. And then if we go to the SEM and get an EDS spectrum of the same particle, we can start to see the elemental composition of that material. The sulfur, carbon, and oxygen are typical of a vulcanized rubber. Zinc could be a filler or a processing agent, and the calcium and titanium are pigments and fillers, probably calcium carbonate. By IR, we see something. This is just rolling it out, not doing any extra work. We get an IR spectrum that's consistent with latex. We start to see contributions from the calcium carbonate, And if we wanted to get rid of that calcium carbonate, we could pyrolyze a small sample of the rubber and get an IR spectrum as well and further characterize it. These particles present as transparent. By polarized light with a single polarizer, we start to see the surface, the fracture surface that's typical of a conchoidal fracture, which is a glassy fracture. So this could be a glass. If we cross the polarizers, oops, looks like I lost that slide. If we cross the polarizers, that whole field would go black since glass has a single refractive index. It's not birefringent. So that would clue us in right away. The SEM data on the same particle shows sodium, a little bit of magnesium, aluminum, silicon, potassium, and oxygen, which is very typical for a soda lime glass. And then by IR, just to complete the cycle, we see that we have a silicon oxygen type of material. This is by attenuated total internal reflectance with an ATR objective on the IR microscope. So we still have comprehensive data. Not all of it's necessary to do, but for demonstration purposes, I think it's useful. Another particle or set of particles which present as, they look like they're shredded, and at higher magnification using one polarizer with a polarized light microscope, we see that it has a tortuous life. So this has been torn, shredded, tells us a little bit about the process it's been through. It's a little beat up. If we cross the polarizers, we see that the particles are now bright. They show these nice interference colors. It's birefringent. We know it has more than one refractive index. The SEM helps us a little bit. We start to see the same shape, the same tortuous appearance. It's shredded. We don't see any fillers associated with it, but we do see carbon, oxygen, and nitrogen. The nitrogen can be helpful to correlate the FTIR spectra, which tells us it's a nylon. So with an amide, we'd expect to see a little bit of nitrogen in the EDS spectrum. So all these, again, all these different techniques help correlate and complement each other as we go through the process. We'll talk a little bit about some opaque particles or some bright particles when they reflect light. By polarized light, they're opaque. Cross polarizers doesn't help us much. It's more opaque since we can't get any light through it. By scanning electron microscopy, we start to see some of the surface features of the particles. The upper right is the entire piece. Lower left is a magnified area of the upper part. And we can see that it's been heavily machined. It's been beat. It has striations. It's been through a process where it's been beaten and machined and stretched and basically beat up quite a bit. If we isolate an area, we start to see some contrast, some different contrast and brightness, which tells me in this particular imaging mode in the SEM that we may have different types of materials. So we'll look at areas 1 and 2 in that lower image by EDS. Area 1 looks like plain old steel, mostly iron. In area 2, we see mostly nickel. So we may have a nickel alloy, we may have a stainless steel, we may have a nickel-plated iron, a nickel-plated stainless steel, which would be odd. But at this point, because the particle's so heavily machined, we really don't know. But we do know we have these elements present. Looking at another particle from the same batch, we also see that we do have some stainless steel, that are high chromium steel associated with it. Again, heavily machined, a lot of striations, telling us that it's had a very torturous path to get to the point where it was actually collected for analysis. This material has an interesting shape in that it presents as two different colors. We have a clear and we have a translucent appearance to it. Polarized light microscopy helps us a little bit. We start to see that there may be two phases here, two different materials. A clear phase towards the outside of the particle and then a more textured phase, which is actually scattering the light, making it appear brown. If we cross the polarizers, again, that clear phase starts showing up as birefringent. It's a little bit brighter. But when we put it in the SEM, we start to see that we indeed have two phases, and what we're looking at is the atomic number contrast. Just the fluorine-rich phase, where the pointer is at 1, shows up much brighter than the carbon-rich phase. So these two materials are easily distinguished using backscattered electrons in the SEM. And if we look at the other phase, it's primarily carbon. FTIR spectra from each material. The clear phase, or the darker phase in the previous image, is polypropylene, and the whiter phase, or the brighter phase in the previous SEM image, is a fluorocarbon, fluoropolymer. Sometimes we find details that aren't always of interest to our customers, but they are to us. We were asked in this particular case to look at a multi-layer foil laminate. It's a packaging material, and it was 3 coatings, or 3 regions that were of interest, and those are 1, 5, and 7 in the image. A razor cut didn't help much. We could tell that there was multiple layers, but we couldn't tell how many layers. What we did in this case is we ion mill polished it with an ion mill polisher. It's a cross-section polisher, which is very good for hard and soft materials. Layer 3 is the aluminum foil that's part of this multi-layer foil packaging, or multi-layered laminate packaging. And now we have layers 2, 4, and 6, which show up a little bit better in dark field. Again, this is a reflected dark field image of the same material that's been ion mill polished. By FTIR, we were able to determine the composition of layer 1, layer 5, and layer 7 because they're large enough. But by FTIR, our limit with the instrument we have is about 25 micrometers in diameter. So we went to confocal Raman microscopy to determine the composition of layers 2, 4, and 6. Ming was able to map it to show that they were different compositions. And if you look at the scale, if I can get my cursor back over here, half of this scale is 25, half again is 12.5. So this layer 6 is probably about 5 or 6 micrometers, and that's well below the resolution of any FTIR microscope. But with the Raman system, we were easily able to get Raman spectra on each layer. So a very powerful technique. But again, this is preceded by the right type of sample preparation to get this information. So for a single particle, or a single particle type, we like to perform at least 3 tests, that being the SEM analysis, the IR/Raman, or the molecular spectroscopy, and the polarized light microscopy, so that we can compare results from each technique and have some quality control over our analysis. If the particle's large, subsamples can be obtained. We can cut them into smaller bits, and then all these tests can be done simultaneously, which is for us a great advantage in time of analysis. So these things can be going on all at the same time. Another example of an aggregate, this material was found on parked cars. When it was wiped off or brushed off or washed off, it actually abraded the paint. Inside of that aggregate were glass fibers, glass spheres, quartz particles, and by scanning electron microscopy, you can see there's actually brighter particles, which are iron particles. There's aluminum metal particles, as well as the quartz in the glass fibers and spheres. So it's a very hard sample, which understandably, if it was rubbed across a car's paint, would cause problems. We got a reference sample. We noticed that the resin holding all this stuff together was acetone soluble. So we're able to do an extraction, compare the 2 resins, the reference and the unknown from the cars, and show that it was a phenolic resin. The phenolic resin, in combination with all the other materials, pointed to an industry that did aluminum casting. so that the vapors from the casting process, when they poured the molten aluminum into these sand molds that were coated with the phenolic resin release agent, was condensing outside of the air handling system and dropping onto cars in the parking lot. A classic example of contamination, which was presented initially as black spots, turned out to be these flies that were embedded either on or in the stoppers. The question was, are they in the stoppers? Were the stoppers purchased this way? Was this happening when the bottles or vials were being filled? Higher magnification with the SEM showed us that in these areas up here, you can actually still see some of the wing structure, and the fly body is embedded in the rubber, and this suggests that the flies were present during the compounding of the actual vial stoppers, suggesting that the flies were present in the vials when they were, or excuse me, in the stoppers when they were purchased, and not during the bottling operation. Dissolution, we have aggregates on the left and on the right. The one on the left dissolved as it should. The one on the right dissolved very slowly. We're able to show just with a couple of pictures that the particle size on the left was very small. The particle size on the right was much larger. So they had very different rates of dissolution. That picture alone was enough for the customer to make some modifications. The polarized light image showing the nice colors in the birefringent particles. The question here was, we were getting aggregates of particles. What were the aggregates? How were they forming? What did they look like? Polarized light started to show some indication that the particles were intergrown, and then looking at them with the scanning electron microscope at higher magnification, we actually saw that the particles were growing through each other. So it was not just a bunch of single crystals that were agglomerating, but it was actually aggregates that were intergrown, and this led to some changes in the way that those products were formed. Occasionally we're asked how things are bonded together. There was two different polymers. It was not apparent to the customer that there was a bonding layer. Just with a quick simple hand section, we were able to demonstrate that there was a bonding layer and then provide them with information as to what that bonding layer was. Another situation where a packaging was being compromised at a facility, and different tools apparently were being used to compromise the packaging. So we were asked to use different types of tools that were supplied and tell if we can tell the difference between a scissor cut or a punch or a tear, and we were able to demonstrate that by using polarized light microscopy, that indeed the markings and the resulting injury to the packaging demonstrated different types of techniques and tools. If we have a bottle of wine on the left that's been consumed, it's one of these single serve, and a bottle on the right that's brand new. The customer, after drinking the glass of wine, was a little concerned that they had these particles in the wine bottle left over. We were asked to isolate and identify the material, or characterize the material. Once we isolated it, we ran an infrared spectrum on it, as well as looked at it by polarized light and SEM. And it looks like the IRR was the most helpful, suggesting that it's a potassium bitartrate or baking powder type material, which is a common byproduct of the winemaking process. It's harmless, but it's certainly not something you want to see in your wine after you drink it. I think this is my final set. We had a situation where we had this protein powder, and an individual was taking it, and they were testing positive for steroids. So the question was, well, the manufacturer is not putting steroids into their protein powder. One, that would be not cost effective, and two, it wouldn't be proper. So it's suspected that someone was putting this material in the protein powder. Sending the bulk protein powder out for analysis for steroids was testing negative, and that's because at the individual particles, shown here as dark particles with white centers, that actually represented the steroid compound were not that common in the bulk of the protein powder. We were able to sit with a stereo microscope and isolate several milligrams of these darker particles, send them off for analysis for steroids, which came back positive, identified the steroid and steroids present, and then go back and demonstrate that, yes, indeed, these were present in the protein powder and they're not typical of the particle types in the protein powder. Higher magnification view there. A bit tedious, but it definitely helped the sensitivity of the process. I'd just like to remind everyone that we are accredited to ISO 17025 by the American Association for Laboratory Accreditation. We are CGMP compliant. We are DEA licensed for Schedule 1 through 4 materials, and we are FDA registered. I believe that's all I have. I thank you for your attention. And I have my contact information up here. And if you have any questions, I think Ming has a few questions he'd like to ask you and try to get some responses.
Hi, Rich. Thanks for the great presentation. Before we open up the floor for questions, we're going to do a quick survey. If you would answer those questions for me, then we can start with the questions. Can you guys see the question screen? We got 90% voted. Just a couple more. Okay, great. I'm going to share the result real quick. So it looks like 84% of you have in-house capabilities for particle characterization and 30% for outside testing. I'm going to hand over to the next question. So what's the most important factor you consider when working with a contract lab? Just waiting for a couple more. Okay, great. Share the result. Looks like quality of result and turnaround time are the two biggest concerns that people have. That's good. And now, final question that I have on here is, what analytical techniques are you interested in? This is a pretty even split with scanning electron microscopy up 70%. Okay, great. Thank you for answering these questions. Now we're going to open up the floor for questions. So please type in your question in the box and we'll address them as much as we can. We have a question, Rich. What is large in your world?
What is large in my world? I would say a millimeter. If I have a millimeter size piece of material, we can do simultaneous tests and have enough left over if some other test should come along in the future.
Okay, great. And How do you make certain that cleaning of the sample does not interfere with, say, FTIR?
That's a good question. It's sample dependent. Everything we do, we're actually monitoring through a stereo microscope while we're doing the analysis. So if we use a particular solvent, we'll run a blank to make sure that the solvent doesn't contribute anything to the IR spectrum. We'll also, hopefully, have enough material to try a number of different solvents if we're trying to either clean or isolate a component that's soluble in the solvent. So essentially it's practice, being mindful of what you're working with, working in a clean area and working with clean solvents and materials.
Okay, and we have a couple of questions about particle size. What is the smallest particle you can do with the micro-Raman system?
Well, the manufacturer claims 200 nanometers, but I think a realistic lower limit would probably be half a micrometer.
The next question is, what does FDA register mean?
That's a good question. FDA registered means that we have notified the FDA that we are available for them to come visit us anytime they'd like to. So they come into our facility, they audit us, they give suggestions on how certain processes are carried out as far as file handling, file storage, testing. Our testing is a little bit unusual in that the type of testing we do, there is no SOP for. We have SOPs for taking a particle through our processes. We have SOPs for the instrumentation, but there is no SOP for an unknown material. Based on what the material presents when you start your examination using the stereo microscope, that leads you as to what analytical tools you'll use and the sequence. So essentially, we've just agreed to work with the FDA and open up our doors for them to audit and inspect our facility at any time.
Okay, and do you have XRD analysis?
We do not have XRD analysis on site. We do have other vendors we work with for that.
One question that just came in is, have you been audited by the FDA?
We've been audited, I think, 3 or 4 times by the FDA.
Okay, great. Do you guys have any more questions? If not, then we thank you for attending. It's been really fun, and we look forward to doing this again. We will be sending out a copy of the presentation. If you'd like to receive a copy, please email me or email Rich. And if this is it, if I don't have any more questions, then actually one question just came in. Have you ever had to go to court to defend your result?
When I first started working for the crime lab, that was a common occurrence. It's not so common now, but I do go to court to defend my results, yes.
One more question. How do you isolate particles?
That depends on the particle. There's a number of different techniques. If it's in a material that's water-based, we can filter it. If it's in an aqueous media, it's usually not a problem. If it's in a more aggressive type of solvent, then we have to have other solvents to isolate it. We can hand pick it using a tungsten needle, some of the techniques that are still taught by the Crone Research Institute. Or we can basically just hand section a material and then use either tungsten needles or a combination of solvents to isolate it. It all depends on what it is and where we're trying to go with the analysis.
Thank you, Rich. And I think this is it. Then we look forward to seeing you guys next time. Please give us your feedback on our follow email as well. Thank you. Bye-bye. Thank you, Rich.